so this is just a quick introduction to the AQA A-Level Biology Spec, something you should really be looking at before the exams to understand what you should be revising, and actually when you go into the exams feeling confident that you covered all the content and know which questions could be asked. So when you first just put AQA A-Level Biology Spec into your browser, you'll get this. And here, first thing to look at is the subject content. This will break down everything that you need to know for your exams. So if you're doing an A-level paper, you can see here 5, 6, 7, 8 are outlined as A-level only, meaning they won't appear in the AS paper. So if you're doing your first year exams, you probably only be assessed on content 1 to 4. But if you're doing the A-level, obviously you need to know all of this content here. If you scroll down, you can see the AS assessments. On paper 1, it will tell you that topics 1 to 4 are assessed. So you need to know about biological molecules, cells, organisms exchange substances with their environment, and genetic information, variation and relationship between organisms. Here you can see the timing of the paper, the number of marks, and actually the percentage of your grade that it will count for. It's important also to look at the question style, so you've got some short answer questions and some comprehension questions. So with the comprehension questions, you're going to really need to apply your knowledge to different situations. So as you can see at AS level, paper 1 and paper 2 both contain topics 1 to 4. Both are written exams in an hour and a half, 75 marks and 50%. The A level, again, containing the same proportion of short answer and extended answer questions. At A level, you've got topics 1 to 4 on paper 1, so similar to the AS paper. Over at paper 2, you start being assessed on topics 5 to 8. And paper 3, you could be assessed on anything from topics 1 to 8. So it's really important to know when you're going into your paper what topics could actually appear on the paper. As you can see, an important thing to know about paper 3 is the 25 marker at the end. This is where you have your essay. You're given 2 titles and you get to choose which title you want to write about. It's really important when you see this essay to start planning and not just dive in, because some of the topics sound really interesting. You feel like you've got a lot to write about, but when you start putting it down on paper, you realise actually you've come to a dead end and you can't actually formulate a 25 marker for it. So it's really important to sit there, think for a minute, and then actually dive into your answer. So at the bottom of the page, you've got your subject content here, and this is going to break down each of the topics we saw earlier to show you which key areas could appear. So here on our subject content, and first of all we're going to look at biological molecules. So this is just a brief introduction here, outlines what the topic's going to be about. So you can see you've got carbohydrates, lipids, proteins, nucleic acids and water. Here under the subtitle monomers and polymers, this is the content that you could be assessed on. So you've got monomers, polymers, monosaccharides. These are definitions that you will need to know. So I recommend you could put it on a flashcard and then put the definition on the other side of the flashcard to really understand these key terms. Again, scrolling down, we've got some images here. These images could be assessed. You could be asked to recognise them or draw them yourself. So again, we're putting these on flashcards to really remember them. On the other side here, you've got opportunities for skills development. This is things that your teacher could assess you on in class, or you could do practicals together looking at these areas. So it is possible that a question may ask about how you could design a practical to look at this stuff. So you could use chromatography or dilution series. So a question may ask you, how could chromatography be used to assess this, or which methods could you use? So understanding all of these areas is really important. Scrolling down again, we've got another opportunity for a development. And here, students should be able to. So you need to be able to recognise from diagrams saturated and unsaturated fatty acids. Another thing you could be putting on flashcards. And explaining the difference of properties between triglycerides and phospholipids. So you could be making tables comparing the two and keeping that to hand so you can look back and remember the differences between the two. Another image that you'll need to know, this is an image of an amino acid. General structure, so recognising that and being able to recreate it in the exam. So here we've got to proteins and enzymes and our first required practical. So you will have done this practical with your teachers, but it's important to be able to recreate it in the exam conditions, be able to write what you did, what you would change, or understand when a question is given outlining this, the results that you would expect and abnormal results and why they may occur. With regard to the required practical, if you look here on the side of your screen, you've got the A-level practical assessment and the AS practical assessment. If you click through here we've got to the A-level practical assessment page. It's got use of apparatus and techniques. So this list here is all the apparatus and techniques you need to use, and it's important to look on the side here. We've got these references that are used further down for each practical. So here's some of the apparatus and techniques. So sampling techniques and fieldworks, you're going to need to remember that for your ecology. Plant and animal responses and physiological functions, so model organisms, having a look at that and when each organism would be best to be used. Light microscope, looking at how to use it, the structure of a light microscope, labelling it, another key area to look at. Here's the required practical activities. So you've got required practicals 1 through 12, and this is the reference here to know which key techniques and apparatus to be using. You need to be able to understand the technique for each required practical, and you may be asked to write how to perform the practical in the exam, or look at someone else who's performed the practical and see what results you would expect, how you would change their procedure, if there's steps missing, maybe fill in the steps, and also be asked to look at the data and formulate tables or graphs, or take information from the graphs and say what it means in a biological context. And this is shown here if you scroll down, use of application of scientific methods and practices. So presenting the data, being able to draw graphs, labeling your axes, using the correct scales, titles, etc. Identifying variables, so you need to know your control variable, independent variable, dependent variables. And again, plotting and interpreting graphs, considering margins of error, accuracy and precision. Learn the difference between accuracy and precision. Put that on a flashcard and keep it in your head. So that's the main thing to be looking at for practicals. So I hope that has been useful. It's only a quick overview of the spec. Please keep using the spec before your exams. Have a look at what you need to be doing, and fill yourself with confidence going into the exam knowing that you've looked at everything that could come up.